Welcome to Chapter 7, everybody. This chapter is all about basic molecular genetics. So we'll be talking about what DNA is and the functions that DNA hold. Then we'll talk about how we replicate DNA, how we produce proteins from DNA. So what is DNA? DNA, as this slide says, is the blueprints of your life. DNA holds the genetic material that makes you who you are. All DNA is identical. So if you look at bacterial DNA, DNA from a plant, and DNA from a human, you'll notice that it is composed of 4 basic nucleotides: adenine, thymine, guanine, and cytosine. Those 4 nucleotides code for every single amino acid that makes up all the proteins that make us who we are. And this is just a little joke here. The genome then is our complete genetic set of information. DNA is held in what we call chromosomes. Chromosomes, when we take the chromosomes plus any plasmids that a cell might have, that makes up all of the genetic information that the cell can have, and that would be the complete genome of the organisms. Viruses, which are non-living, sometimes don't have DNA, they only have RNA. But again, viruses are non-living, so they're not part of that living group. So cells that want to multiply have to be able to replicate their DNA before they multiply so that they have a complete set of genetic information to go into the new daughter cells. They also have to be able to produce proteins because you need proteins for replication and for cell division in a nutshell. The central dogma of molecular biology is that DNA codes for RNA, which is what we use to produce proteins. This is what DNA looks like. Notice that DNA runs anti-parallel. So on this side we have a 5' end, on this side we have a 3' end. Over here in the blue strand we have a 5' end, up here is the 3' end. They run anti-parallel, they run in the opposite direction of each other. But also notice that adenine always binds with thymine. Adenine and thymine again. Here, guanine and cytosine, cytosine and guanine. They always bind together because they are complementary to each other. The backbone is composed of deoxyribose and the phosphate, and then the rungs are those nucleotides. I just said all of this. Oh, they're bonded together through hydrogen bonds, which I didn't mention that. This is just a larger picture of the same thing. RNA then doesn't contain ribose or deoxyribose sugar. It contains a ribose sugar. RNA also does not contain thymine. Instead, it has a different nucleotide called uracil, which will bind to the adenines. RNA is always single-stranded, and it's shorter than DNA because it's only coding for proteins. There are 3 types of RNA that are used in protein synthesis. Messenger RNA is what carries the message of the DNA in an RNA form. This is the one we typically talk about when we talk about transcription, but they're all produced via transcription. Ribosomal RNA is what produces the ribosomes, those little tiny structures that produce proteins. When these ribosomes get involved in or get together with certain proteins, they form the ribosome, the large and the small subunit. And then we have transfer RNA. Transfer RNA carries the amino acids to the growing protein strand. In bacteria, DNA synthesis is going to be in a circular direction, because DNA in bacteria is circular. They have one single strand of DNA, double-stranded DNA molecule, and it's circular. And so you're going to have replication occurring in both directions along that DNA strand. There are a lot of different proteins that are involved in replication. I don't usually make people know every single protein, but going through this list, just know the basic function, what they're doing, and then if I do ask a question, you'll know those, okay? So replication of DNA. First, we're going to have to break the hydrogen bonds so that the DNA molecule, or so that the parent strands can be replicated. DNA helicase helps with breaking those bonds, unwinding and unzipping the DNA. Topoisomerase or DNA gyrase also helps with breaking those bonds. Primase is what we use to synthesize an RNA primer. The RNA primer is necessary for DNA polymerase to bind. DNA polymerase binds only to a double-stranded molecule. So we have tiny little RNA primers that bind to the 3' end of the DNA, and then DNA polymerase will bind over the top. So DNA polymerase then basically will bind at the RNA primer to absorb or break down the RNA primer and then start producing a new daughter strand of DNA that's complementary to the parent strand. This is going to occur on both sides of the DNA strand, so on each of the parent strands. And so you can see here the DNA polymerase. So see here how it's a 3' end. DNA polymerase can bind right at the very top and can start synthesizing in a 5' to 3' direction along this DNA strand since it's a 3' strand. But since the blue strand here is the 5' end, we can only bind on the 3' end. So as the DNA molecule is opening, we're going to bind an RNA primer and then a DNA polymerase and produce a small section or segment of newly synthesized DNA. And then again, we have RNA primer and then a DNA polymerase. And we're going to synthesize another section. And then again, an RNA primer and a DNA polymerase. And we're going to, RNA primer, here's the primase, and then the DNA polymerase will bind here, and we'll get another segment of daughter strand produced. Then DNA ligase will come along and seal everything together. This is the lagging strand, this is the leading strand. So in bacteria, again, replication is bidirectional. So it occurs on both parent strands, but they're going to run in opposite directions. And when we're done, we'll have two completely identical DNA strands, circular DNA strands, each containing one parent and one daughter strand. And so here you're going to watch DNA at the replication fork, okay? Maybe, if I can get this to work. There we go. A lot of stuff going on my computer.
The helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3'
continuously in the 5' to 3' direction.
One strand of the DNA,
Synthesis
called
of
the
the
leading
complementary strand,
proceeds
called the
continuously
lagging
in the
strand,
5'
is more
to
complex.
3' direction.
Polymerase can add new
Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading
And remember
strand is
that those
synthesized
little
continuously,
discontinuous
while the lagging strand is
segments of
synthesized
DNA on the
discontinuously.
lagging strand are called Okazaki fragments. And here you can see bidirectional replication. Much better than I can actually do it. So I can't draw this. So
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
Right there. Okay, and so here's my replication, kind of showing my DNA will always be double-stranded and straight because that's just how I do it. So here we have it. It's already been unwound and unzipped. So we're going to have DNA polymerase come along, and then, oops, the nucleotides come in, the complementary nucleotides. And when we're done, we have two identical DNA strands, each consisting of one parent and one daughter strand of DNA. So DNA replication takes takes a certain amount of time. In E. coli, this takes approximately 40 minutes, but bacteria like E. coli replicate every 20 minutes. And so before one replication of their DNA is even complete, each of the strands, the newly synthesized strand as well as the parent strand, is already undergoing another set of replication. So by the time the cell divides, each of the chromosomes is already halfway through their replication process. Transcription then is the production of a message of DNA in an RNA form that is usable by the cells. Cells can't use DNA, and there's a lot of, discussion as to why the cells can't just use DNA straight up, but one of the hypotheses is that the cells can't recognize thymine because thymine is not in RNA, so it could be that the thymine is one of the factors that inhibits DNA from being able to be used directly. But whatever the case is, our cells can't recognize DNA. They do recognize RNA and can use the RNA to produce proteins. For transcription to occur, we use RNA polymerase, which is going to bind to a region on the DNA called the promoter site. and then it's going to synthesize a new strand, or a strand that is identical to the coding region of the DNA, and it'll be in an RNA format. So each strand of DNA can be the coding strand, or can code for a protein. Whichever strand we're using as the coding strand, the other strand is called the template strand, or the minus strand. We use the template to produce our copy of the coding strand. So if you look here, here's your plus, or your coding strand of DNA. This is the template. So we use the template to produce a copy of messenger RNA. And if you look at the messenger RNA molecule, it is identical to this strand. Where you see a G here, you see a G here. Where you see a C here, you see a C here. The only difference is where there's T's up here, you have U's down here. The entire molecule is identical otherwise. In bacterial cells, their messenger RNA can actually contain multiple proteins. So you can be coding for more than one protein. That's called polycistronic messenger RNA. In eukaryotes, we typically produce monocistronic messenger RNA. And there's a reason for that. We're unable to code for two proteins at the same time. Our transcription is a lot more complex than bacterial transcription. So here you can see the RNA polymerase binding to the DNA at the region called the promoter site. RNA polymerase is going to run along the DNA molecule. As it moves, it denatures those bonds between the double-stranded DNA, allowing RNA primers or RNA nucleotides to come in and produce a copy of the DNA in an RNA format. And this is going to occur until we hit the terminator sequence. Once we hit the terminator sequence, then the RNA is free to be translated. The DNA will wind back up, and the RNA polymerase can be used again. The promoter region will determine which direction DNA or RNA polymerase will replicate, so which way they're running. And this allows you to produce lots of different RNAs at any given time. So you have a lot of different promoter regions that say, okay, produce this side or this side. And you can have multiple RNAs binding and producing the same messenger RNA molecule. So here's my transcription that I made up. Minus strand, plus strand, so coding strand template. There's the promoter and the leader sequence. Oh, sorry. And here's the messenger RNA molecule right there, okay?
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
All right, so we're going to stop here and we'll start with translation in the next video.